Gene therapy promises to cure diseases at their genetic roots, but there's a catch. Most delivery systems can only reach the liver, leaving other organs untreatable. NanoPalm has developed AI-designed biorobots, tiny patterned nanoparticles that can travel anywhere in the body to deliver genetic treatments right where they're needed. Ali, let's start with the problem. Why do most gene therapies get trapped in the liver, and how have your biorobots managed to get around that?
Not only gene therapies get trapped in the liver, almost everything we inject into our body get trapped in the liver. And this is how liver works. It will just clear our body from foreign molecules. The current gene therapies has certain charges to them. They call them positively charged lipids. It will be easily cleared by the liver. To go around that is not only you change the charge, but also you change the architecture. Today's gene therapies are spherical in nature, so you have to deviate from this shape to go from spherical to nanospherical. If you change the shape of lipid nanoparticles, you can actually escape liver.
You call them biorobots. It sounds futuristic. What are they exactly, and what makes them different from traditional nanoparticles?
Biorobots are made of biological molecules. The term bio comes from the makeup of those robots. And also, they are more like soft materials as opposed to the perception of what a robot is, mechanical robot. Those are more biorobots.
Your first target is sickle cell disease, which is especially prevalent in the Middle East and Africa. How is your treatment designed for those patients?
Today, therapy is called ex vivo, meaning they need to take the bone marrow of a patient and then take it to the lab. And then in the lab, in a petri dish, they do the gene correction. And now those cells are free of sickle cell disease, but they need to be transfused back into the patients. So this is a long supply chain and expensive. What we do is we actually inject directly into the body. And that's the transformation here from what's called ex vivo to in vivo. And that's what makes us different.
You've built a library of over 800,000 nanoparticles that teach the AI. How does having that scale of data give you an edge?
What's public out there are a bit random, disordered lipid structures. So those lipid nanoparticles that are out there predominantly of the same shape and same size and probably similar chemical composition. You cannot train AI on this type of data. AI will need different shapes, will need different sizes, will need different chemical compositions, and so on. You need the other hundreds different features. If you have them all in your hand, then your data become really unique to what's public there, and you will have a competitive edge because now some of those data does not exist in nature, others are not publicly available. Then the AI model will learn on really unique data and private data. That's one thing. The other thing is the data out there is not solving a problem in the field of lipid nanoparticles. Lipid nanoparticles are often unstable in structure, designed with toxic components, and they cannot target specific organs. So we had to create our own data to while solving this problem. Now the AI can see, can tell you which one will target which organ, will tell you if it's toxic or safe, it will tell you if it's stable or not ahead of time.
From a business perspective, how do your partnerships work? What's the model for turning these discoveries into commercial therapies?
Our business model is co-development. Basically, our pharma partners can provide us with their drug, and combined with our cure delivery platform, we create new possibilities, and we create shared success between our partner and us. So our business model is literally we both co-develop drugs and we create new possibilities.